We also saw that when we looked at the different feed-in pathways, like if it's not glucose, if it's mannose, or if it's fructose or something else, right? We need to find ways to get things in and out. If it's glycogen, we need to find ways to get things in and out. Right? So we've now hit a number of different ways to do that. So one good way to kind of synthesize all this material for yourself is to kind of just, have a piece of paper over here somewhere and kind of like add things to it as we go through so that you see, how things fit together. So where we're going now is we've got an overall view of energy yielding pathways, so catabolism. Right? And what this is showing you here, this sort of blue part here, is where you're getting most of your ATP out of it. E minus, it doesn't mean there's free electrons flying around, it means that we've reduced cofactors. NAD, FAD, for the most part, just those two. Carbon, we've gone from 6 to 3, of course 2 of these. Then we go to 2 in this complex that we're going to study next, this pyruvate dehydrogenase complex. The 2-carbon compound that results linked to a cofactor called coenzyme A, which has a thioester, which you'll remember as a high energy linkage. That goes into TCA cycle. TCA cycle shoots off CO2 and electrons all over the place. So you get full oxidation of glucose in TCA cycle. So there's nothing left but CO2. Right? And a lot of energy embodied in redox reactions, embodied in high energy redox reduced cofactors that then give them away again. Here are the reduced cofactors, and they're giving their electrons away to make ATP. And where the electrons ultimately end up, they end up in oxygen. What do they make when they add to oxygen? They make water. Right? So this overall picture is something that is clearly very central to what we're talking about. All right, so we're at pyruvate, and we want to go on from there. And in eukaryotes, the citric acid cycle doesn't occur in the cytoplasm like everything up to now has occurred in, but instead it occurs in the mitochondria, so we have to get across. And so diagrammatically, you can see here, this is the mitochondria. Mitochondria has 2 membranes, by the way. Membranes are the bilayers, right, the leaflets. And so there's an aqueous space between the 2 membranes. It's called the periplasmic space. There's also 2 membranes around the whole cell, and so there's a periplasm around the whole cell. But now we're talking about an organelle inside the cell that's membrane bound, and there's 2 membranes around that. And it's very important to know that because we need to keep track of, when we think about these reactions, where we are with respect to the inside of the mitochondria, the periplasmic space, or the cytoplasm. And things are going to be coming from both directions, and certain machines that are doing the work, proteins, are going to be in one place or another, with their active sites pointing one way or another. And so we have to keep track of that kind of biological topology, if you will, however you want to think of it. You got to keep track of that stuff. So for now, we just need to remember that glycolysis in the cytoplasm, pyruvate is translocated into the mitochondria. How does that happen? There's a specific transporter. Okay. So pyruvate is a charged molecule. Notice it's not phosphorylated, but it still has a carboxylate. So typically, charged molecules don't cross membranes easily on their own, membranes being nonpolar. Right? So they require some facilitation. And there's a specific cofactor that allows pyruvate to go in. It does not allow, for example, phosphoenolpyruvate to go in. It allows pyruvate to go in. So then the next steps, PDH is pyruvate dehydrogenase complex. Sometimes it's written as PDH, sometimes written as PDC. I'm afraid I may not have been that consistent, but PDH and PDC are exactly the same thing if you see either one of them. So the matrix of the mitochondria is the aqueous part inside of it, right? Just like the cytoplasm is the aqueous part inside of the double membrane of the whole cell. The aqueous part inside the double membrane of the mitochondria is the matrix. And there's a lot of convoluted membrane structure inside the mitochondria, which is important for function, and we'll talk about that later. And oxidative phosphorylation occurs in the inner mitochondrial membrane. So oxidative phosphorylation is this step here. This is phosphorylation of ADP. Oxidative, meaning through redox reactions. And so this electron transfer chain is in the inner mitochondrial membrane. One enzyme of the TCA cycle is also in the inner mitochondrial membrane. Okay.
You say in the inner mitochondrial membrane, you mean in between the matrix and the periplasmic space?
Correct. Yeah. So there's 8 enzymes in the TCA cycle. 7 of them are in the matrix of the mitochondria, kind of diffusible enzymes, like glycolysis enzymes are in the cytoplasm. These enzymes are in the matrix. But the 8th one is on the membrane itself, that is to say, membrane associated. And that enzyme is an enzyme that has FAD bound to it. Right? Does that make sense? You can tell me why that particular enzyme with FAD bound to it would be the one in the membrane.
Is it because FAD is not diffusible? So
Yeah, right. So if you have an enzyme that's working, a redox enzyme that's working through FAD, it gets reduced with your fuel molecule, which is the sugar skeleton going through the TCA cycle. And that fuel molecule has electrons to donate to FAD. Now you have FADH2. Well, great. If you have FADH2 on your protein and it can't leave, right? Because it's not diffusible, then now you just lost those electrons. So you have to there has to be a way to get those FADH2 electrons into the rest of the electron transport chain. And that whole rest of the chain is also in the inner membrane. So that's going to be the hand-on. The other enzymes in the TCA cycle are going to generate NADH, and that is diffusible. So NADH is diffusible whether it's in the mitochondrial matrix or in the cytoplasm. Right? So the enzyme that does this is pyruvate dehydrogenase complex. And this is the overall reaction. This is the overall standard state delta G. It's just like one more step in the glycolysis, like the next step after glycolysis. But this enzyme is a monster, and it uses 5 different cofactors. Okay, it's an oxidative decarboxylation, so the kind of reaction that was in the pentose phosphate pathway to go from 6 to 5 is now going from 3 to 2. The cofactors that it's using, 3 of them we've now looked at. NAD, you know about, FAD, you know about, and TPP, thiamine pyrophosphate, you also know about. We just talked about it in the context of pyruvate decarboxylase. So the other 2 cofactors we haven't seen yet. One of them is called lipoic acid, or lipoate, and the other one is called coenzyme A. And coenzyme A is the one with the thioester, and this is what that one looks like. Okay. It's got an adenine ring, got a sugar, 2' OH, another little RNA-derived cofactor, or RNA-related cofactor. It's got carrying a phosphate there, bisphosphate off the 5' end, a linkage here, complicated structure here called pantothenic acid. Another, by the way, another B vitamin. This is where your B vitamins are going if you take them. And then the business end here is beta-mercaptophentylamine, and the end has a reactive thiol group. And this thiol group is going to form a thioester, which is going to be a high energy compound, and it's going to form that with the 2 carbons that are left over after the CO2 of pyruvate goes bye-bye in the earlier, in one aspect of this reaction. Okay, so CO2 is going to be split off, here it is, right? And coenzyme A, this whole business here it is, right? CoA, S, carbon, so thioester, and then a methyl group.